This lecture will be on bioavailability and bioequivalence. This is going to be really important for pharmacists to understand because it's directly related to whether or not a drug can be substituted for another drug and be considered, you know, uh, basically equivalent therapeutically or clinically. So there's actually many different levels of equivalence that we'll talk about, especially at the end, and how drugs are compared to each other. So the learning objectives are to define bioavailability, bioequivalence, and drug product performance, explain why certain drugs and drug products have low bioavailability, explain why first pass effect as well as chemical instability of a drug can result in low relative bioavailability, distinguish between bioavailability and bioequivalence, explain why relative bioavailability may have values greater than 100%, describe various methods for measuring bioavailability and the advantages and disadvantages of each, and something you would have never seen before probably is to describe the statistical criteria for bioequivalence in 90% confidence intervals. Okay, and then compare and contrast pharmaceutical equivalence, pharmaceutical alternatives, and therapeutic equivalence. First, drug product performance, bioavailability, and bioequivalence. Drug product performance is a release of drug substance from the drug product leading to bioavailability of the drug substance. Okay, so it's mostly about the release of the drug from the dosage form. Bioavailability is the rate and extent to which the active ingredient is absorbed from a drug product and becomes available at the site of action. Sometimes you'll hear the term absolute bioavailability, and that means the percent absorbed. If you're just referring to plain bioavailability, that's the rate and the extent to which the ingredient is absorbed, the active ingredient. Bioequivalence is the absence of a significant difference in the rate and extent to which the active ingredient becomes available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. So bioequivalence means two drugs are basically having the same bioavailability. So first of all, what about bioequivalence when new drugs are being developed? Right, a brand new drug that some company wants to market, they have to submit a new drug application, which you might be familiar with already. So that's called an NDA, again for a proprietary or brand name drug. Bioequivalence studies are still necessary, believe it or not, even though we're talking about a new drug. So early on in a clinical trial, they might have a different formulation than later on in the clinical trial, and they have to show whether or not those are bioequivalent. Formulations used in clinical trials in stability studies, if different. Okay, so sometimes when they're studying the active ingredient stability in different situations like different pHs, different temperatures, the formulation might be different than that's used in the clinical trial. Okay, so they have to basically show whether or not those are bioequivalent. Clinical trial formulation versus final to be marketed formulations. Believe it or not, the clinical trial formulation used might have slight, sometimes maybe more dramatic differences, but usually slight differences from what's finally going to be marketed. So there might be some final tweaks or changes in the formulation after the clinical trial to improve some parameter. So that they have to show whether or not those are bioequivalent. Okay, because let me put it this way, if the final formulation is not bioequivalent to the clinical trial formulation, that raises a serious issue because you would question whether or not the final formulation would result in the same outcomes, right, as the clinical trial formulation, because essentially, right, there might be a difference in the Cmax, there might be a difference in the area under the curve, and that can affect the actual clinical outcome. New formulations of patented products still need bioequivalence studies. So, for example, if there's a new tablet combining active ingredients, so if some company has a patent on an active ingredient, so the brand name drug, but then they want to combine it with another drug and also market that and patent that, and market that exclusively. Because they're combining it with another agent, they have to prove whether or not it's bioequivalent to the original drug, the individual drug. And if it's not, in most cases, they'll have to do more studies on the clinical effect, right? If it's not bioequivalent, most likely they're going to have to do another clinical trial to prove that it's, again, safe and effective. So this is the whole kind of flow chart of why the original drug manufacturer may still need bioequivalent studies. So first of all, we have an active pharmaceutical ingredient. That's what they're starting with. That goes into the clinical drug product. Okay, they do clinical efficacy and safety studies, pharmacokinetic and bioavailability studies. But then, as I mentioned, the marketed product may be somewhat different in formulation than the product that they use in the clinical trials. Okay, so they have to at least prove that the dissolution profile is similar or the same, and or bioequivalent studies have been done showing that this marketed product is bioequivalent to the clinical drug product. Okay, and then after it's on the market, any changes to a formulation, for example, if one of the excipients is in short supply and they wanted to change that to one that's readily available in the world, right? A new excipient, they have to prove that that excipient does not interfere with the bioavailability, right? So it's really stringent. A lot of the lay people out there don't realize how stringent these rules are for basically getting a product out there, even if it's you're the brand name manufacturer and you wanted to make a small modification. Okay, once in a while they will accept non-clinical bioavailability studies, so in vitro tests. We'll talk about those later on. So why would something be allowed to basically prove bioequivalence just by doing a dissolution test or something like that, rather than an actual live patient absorption study? And like I said, we'll talk about that. But that's ultimately always up to the FDA. That's the Center for Drug Evaluation and Research, or CDER. So some of the post-approval changes requiring bioequivalent studies would be if you change the supplier of an active ingredient. And the reason why that's critical is because, as you're probably all aware at this point, the source of the active ingredient can have serious implications. For example, are there impurities in there that are toxic? Remember, there were a bunch of drugs that showed up over the past year or so. Basically, well, it was probably 2019, 2020. Basically, that showed that there were cancer-causing substances. Might have been in Zantac and things like that. But there were actually several drugs where they showed that, and it turned out to be an impurity. So that might easily be related to the supplier. So how did the supplier produce it? Not only what country, but more importantly, what were the measures they used to ensure that the drug was pure and didn't have any contaminants. Change in the manufacturing process. Any small change can have a rippling effect, right? What kind of blender are you using? How long are you milling it for? Like everything, any little change can have a significant effect in the end. Change in the manufacturing location or site. The site could have different humidity, different air quality, and that's supposed to be controlled, heavily controlled, the air quality in these these manufacturing sites. But anything in the location or site could potentially have a difference in the result, believe it or not, in the quality of the product. What most of us probably think of, well, if you've ever thought of bioequivalence in your life, which you may not have, other than my past few lectures, but bioequivalence for generic drug applications is what we normally think about, which are also considered abbreviated new drug applications, right? That means the active ingredient has already been on the market in some way or another. So there's a brand name product, and some other company, now that the patent has expired, some other company wants to come out with a product, so they have to submit an abbreviated new drug application. One of the most important things at this point is to prove bioavailability or bioequivalence, right? So bioavailability should be the same. because the active ingredient has already been proven to be safe and effective. So the most important thing is to prove that it's absorbed at the same rate and the same extent. Okay. So that would be, again, the first bullet that's already covered by previous studies. So we're focusing on bioavailability. These tests are generally performed in healthy humans under fasted and fed conditions. So you have to realize that, and hopefully that usually doesn't make a difference, but even though the drug's intended to treat a disease or an ailment, most of the bioavailability studies are done in healthy humans. So whether or not that actually causes a confounding effect or causes a problem, I guess it's going to really be something you wouldn't know until the drug got on the market, unfortunately. So there are instances, but I don't have specific examples, but I can imagine instances where a disease might make the bioavailability very different compared to a healthy human. So again, the FDA can change the rules. They can add more rigorous stipulations that maybe you cannot test it in healthy humans, particularly if the drug is very toxic, right? Like a cancer drug. Sometimes, once in a while, dissolution profiles may be sufficient. That would be more ideal for the biopharmaceutical classification system, or BCS, high solubility and high permeability. That means the drug dissolves very well. It's absorbed basically like 100%. So you're pretty sure that as long as you have the correct amount of active ingredient in the formulation, it should be absorbed rapidly and to the same extent. And that's not super common, but it does happen where, again, you don't have to even do a clinical study on absorption. So the generic manufacturer always needs bioequivalence studies. Remember, the brand name manufacturer would need a bioequivalence study only if they're changing the product. So again, you have the active pharmaceutical ingredient that goes into the generic drug product. They have to do either comparative dissolution profiles or bioequivalence studies in actual humans. Okay. And then basically, if in a generic drug, generic drug manufacturer they can change the formulation after it's approved, but then they have to prove bioequivalence again and again and again. So anytime you change the product, you have to prove bioequivalence. So I'm going to mention this Orange Book, and we're going to come back to it also later. But every single generic product has to be compared to something else that's already in the market, and usually it's the brand name product, right? It's the first one that came out. And the reference drugs, again, that all the other drugs are compared to for bioavailability or bioequivalence, the reference drugs are listed in something called the Orange Book, which probably used to literally be orange, and now it's just an online database for the most part. Again, it's usually the original brand name or a marketed drug that is the reference drug. Reference listed drugs corresponding to generic versions are listed by the FDA in its publication. Technically, it's called the approved drug products with therapeutic equivalence evaluations. That's the Orange Book. Right, so technically, that's the actual name of it, the long name there. The Orange Book is publicly available and searchable on the FDA's website. I found it in about a minute or less, just going on Google, probably like 20 seconds. So it's really easy to find on the, if you just type in Orange Book FDA. If you really, you never know if you might have to do that actually during your career later on. So just an introduction here again to the Orange Book. So the way it would look in their database online would be you'd have RS for the reference standard. That means all the other drugs, or all the other formulations, I should say, are being compared to this one. So all these other manufacturers had to prove that their product was bioequivalent to this one from, I don't know how to pronounce that. That might be a, I don't know if that's an abbreviation, ATNAHS Pharma, US Limited. So interestingly, if you look at that, that doesn't look like a really big big pharma company that you've heard of probably. At least I haven't. So what'll happen from time to time is if a product is very, very old, it's been on the market for decades, you might find that the brand name manufacturer might not even make it anymore once in a while, right? They might not make it anymore. And this one might actually, and I shouldn't say that because this one, Naprosyn was the original brand name. So maybe that was actually the original company who made it. But regardless, sometimes the reference standard might not be the original brand name manufacturer. It might be, if the brand name manufacturer discontinued their product, the reference standard would then generally be something else that's still on the market. So in this case, yeah, Naprosyn was always the brand name. So that appears to be the correct original company that made the drug. So all of them are compared to that. Okay, so in this case, it looks like we have different doses here as well. Believe it or not, you can prove bioequivalence even using a different dose if if you do the appropriate calculations, and we'll talk about that. Okay, so that's kind of a nice convenience for the generic drug manufacturers. They can try a different dose and still prove bioequivalence if they do, again, they use the correct equations and things like that. And in the FDA, clearly the FDA will take your data and analyze it themselves using their own software or equations, and not just take your word for it. So you're supposed to be providing generally the raw data to the FDA to make that determination. So how is bioavailability measured? Usually area under the curve, right, it's always a major parameter measured in bioavailability studies because it's proportional to the amount of unchanged drug that reaches the systemic circulation, right? So that's a very kind of detailed description because remember, you're measuring the drug in the plasma in the systemic circulation, usually drawing blood from a vein. So that's drug that's already reached the plasma, right? It's not drug that's just in the intestinal cells. It's not just in the portal vein. It's already gone, through the first pass metabolism if there was going to be any. So it's already done all that, and then it's in the systemic circulation. So that is area under the curve. So you're just measuring the area from 0 time to infinity, right? And there's an equation to do that. Okay, and that's one major way. So AUC has to be pretty similar from product to product if they're going to be equivalent. Usually things like Cmax as well has to be similar, and we'll talk about that as we proceed here. So I actually mentioned this earlier, but here it is in the text. Absolute versus relative bioavailability. Absolute just means very simple. It's what percent of drug is absorbed into the systemic circulation, right? What percent? That's simple, right? Relative means the percent difference in absorption relative to another product. So actually what I mentioned earlier was just what absolute means. I didn't really mention the second one until now. So remember, we talked earlier about just general bioavailability is rate and extent of absorption. Absolute is extent, only the extent, which is the percent absorbed. Relative is very simple. It just means you're comparing one product to another product, right? It means the percent difference in absorption from product to product, right? So we'll talk about that, but that's why if your new product, or your basically your generic product that you just developed has a higher bioavailability than the reference product, the relative bioavailability will be over 100%, right? It might be 105%, 110%. If it's much more than that, we're going to talk about why that might not be bioequivalent at some point, right? What is the threshold where it's really no longer considered substitutable or equivalent? So absolute bioavailability compares the intended administration route with the IV administration route. Okay, in this, it might look a little complicated, but all it's saying is that, right, the only way you know the percent of the drug that is absorbed orally is if you have something to compare it to, and that has to be 100% absorption, which would be the same as giving it IV. So if you give it IV, you see this, and then if you give it orally, you see this, and you can calculate based on what you should see, right, if 100% is absorbed, you can figure out how much was absorbed through the oral route, right? Okay, so absolute bioavailability is the area under the curve of the extravascularly administered drug product divided by the area under the curve of the IV product, right? So that just means, in this case, oral AUC divided by IV AUC, right? So it should always be, think about it, it should always be basically 100% or lower, right? If we convert it to a percentage, right? It should always be 100% or lower, because you can't possibly absorb more than you would with an IV administration, right? And again, it's called absolute because it's compared to 100% absorption. So that just means, right, you're just saying one product at this point. Each single product has their own absolute bioavailability. When we get to relative, that means we're comparing two different products. Okay, that's the equation there that I put in a little too late, but I forgot that I had that animated. But again, it's AUC oral. In this case, if we're talking about an oral product, the black line, AUC oral divided by AUC IV, right? And again, well, I should put it this way, the highest number we're going to get is a 1 here, right? If oral were identical to IV, we would get a 1 here. Okay, and we can't go beyond that. And then if you change it into percent, you just multiply by 100, that'd be 100%. So this is what I was referring to. This equation is really helpful because drug companies can basically try a different dosage and still calculate bioavailability, compared to the reference product. So again, just imagine if a dose given IV is different from the dose given orally, we must correct for this using the equation. Okay, so and remember, absolute bioavailability always requires an IV. At least one of them has to be an IV dose, because you need to know what 100% looks like in terms of area under the curve, get 100% absorption. So you see the fraction absorbed equals AUC of the oral times the dose of the IV divided by AUC of the IV times the dose of the oral. So it might look a little strange at first, but just imagine for a minute if basically if you had if you doubled the oral dose compared to IV, but the area under the curves were equal, that would tell you that somehow that oral dose did not get in 100%. And if it were exactly same AUCs but double the oral dose, that would give you about a 50% bioavailability. So I encourage you to try that with different numbers to see if you get that, and then hopefully it'll make more sense how we can calculate bioavailability with this correction factor even with different doses. So relative bioavailability, remember that's comparing the not IV at all, right? Because IV is always 100%. Relative means relative to the reference product, right? So what is the absorption of your new product or generic product usually compared to the brand name or whatever the reference product is. Okay, so the equation is very similar. Product A would be the new formulation. Okay, so that's the area under the curve there for the new formulation, the dose of the new formulation. Product B would be the reference standard, area under the curve, and dose. For example, if we gave equal doses of each product and AUC-A is higher than AUC-B, we would have fraction, relative fraction absorbed higher than 1. This means drug A is more bioavailable than drug B. So again, if the AUC-A were higher and everything else, like the two doses were equal, I should say, AUC-A is higher and AUC-B is lower, that would give you a number higher than 1. And if you convert that to a percent, it would be over 100%. So the fraction relative, fraction absorbed relative to the reference product is over 100%. So that's what one of the learning objectives, right? So how can you get a percent higher than 100? Because it is relative. So there's anything can be relatively higher than something else, right? So in terms of people's body weight, in terms of people's speed, their their water intake. I mean, so I'm just saying that's just an example that anything could be higher than if you have a comparison product that's not IV, right? If it's oral, if it's something that doesn't have 100% bioavailability, what I mean is if the reference product has some something lower than perfect bioavailability, then there's a chance that another product might be able to overcome that or be absorbed more than that. So here's a practice problem related to this. So the bioavailability of a new investigational drug was studied in 12 volunteers, for example. Each volunteer received either a single oral tablet containing 200 milligrams of the drug, 5 milliliters of a pure aqueous solution containing 200 milligrams of the drug, or a single IV bolus injection containing 50 milligrams of the drug. So you might immediately realize that, so we have what we need. We have IV. IV is always going to be used to determine absolute bioavailability. So it'll be IV compared to any other route. And then we have two different, we'll just imagine one of these is going to be the reference product. We'll just imagine that, but we have two different products. Okay, one of them is a solution, one of them is an oral solid form, right? So it's a tablet. So we expect generally to have different bioavailability in some cases, right? In many cases there. So plasma samples were obtained periodically up to 48 hours after the dose and assayed for drug concentration. The average AUC values are given in the table on the next slide. So of course I have a pause symbol here, but you're going to need to look at that table and then pause. From these data, calculate the relative bioavailability of the drug from the tablet compared to the oral solution, right? And then the absolute bioavailability of the drug from the tablet. So let's go over this one more time. I want to know the relative bioavailability of the drug from the tablet compared to the oral solution. So I want you to assume the oral solution is a reference product, right? And so product A, right, would be the new one that you're testing. We'll just imagine that the tablet is product A on the equation, and the reference product, which is B, would be the oral solution. And then the absolute bioavailability will require you to compare, in this case, the tablet to the IV formulation. So this is the table of data, so please utilize this table, and I'm going to reveal the answer. And, I'll give you a few minutes. Well, you can pause it, right, and give yourself as much time as you need. Okay, so, but just to show you how this works out, or how this is organized, dose in this column, area under the curve here, and you don't have to use a standard deviation now. In fact, I don't have you do a lot of statistics even later, if any, but that's just to show you that this is real data. We're just imagining this is real data and there's variability. Okay, so, and that we will discuss this later on, how variability affects your conclusions to some extent. But again, oral tablet, oral solution, IV bolus, so make sure you're choosing the right row, the right data, and then do that calculation. So you have, again, like 5 seconds to pause it, and then I'll reveal the answer. So please work on that now. 5, 4, 3, 2, 1. Relative bioavailability is 89.5 divided by 86.1. Remember, I said that we are using the, we'll say the oral tablet is we'll just imagine that's the new product, and the oral solution might be the reference product. Okay, so that would be, again, over it's 1.04, so if it's over 1, converting it to a percent just means multiplying it by 100, right? So that's 104%. Okay, so that means the oral tablet had, obviously, you could see here, slightly higher absorption than the oral solution. And you can see that in the area under the curve being a little higher. Now, you might ask, is that realistic? Would the solution really be lower absorption? It depends, right? What if the solution, because it's already dissolved, what if that allows just destruction of the drug molecules in the stomach, whereas the oral tablet might be protecting those drug molecules until they get to the small intestine? So it is possible that this can happen, even though it's not usually common that the solution would have lower. IV bolus injection, then we're going to need that for the next calculation. So that would be 89.5 because again, I said compared to the tablet. So tablet compared to the IV for absolute bioavailability. So that'd be 89.5. And remember, this case the dose is different. In the first case, it was not different. So we didn't really need that in the equation. Okay, you don't actually need that in the equation at all if the dose is the same. If the dose is different, you need to use that equation accordingly. So it's 89.5 times 50. Okay, remember, because on the top you have the opposite. You have the oral tab, in this case the oral tablet AUC, but then the IV bolus dose, right? So the opposite products on the top, and then same thing on the bottom. 37.8 times then the dose though of the oral tablet. That equals 0.592 or 59.2%. That means the oral tablet only has about a 59.2% systemic bioavailability. Obviously, it can never be higher than the IV, and if you ever get a number higher than 100% for absolute bioavailability, there's something very wrong with the calculation. So keep that in mind. So I just have a few questions here related to the last exercise. The first one, of course, in my typical style, I already answered that. So the only reason, one main reason why a liquid might have lower bioavailability is because it might have lower stability of the active ingredient in the gastric fluid because of the acid. Second, are the area under the curves statistically different? And why does that matter? So you'll find out later on that, and we're not going to calculate this now, the p-values or anything like that, and we probably won't even do it at all later on. But what I'll do later on is provide you with that hypothetical data, p-values and confidence intervals and that kind of thing, without actually calculating it, and then we could make our conclusions from that. But you'll see later on that bioequivalence requires that these are statistically not different, and that's not enough. You'll see later on that not only do they have to be statistically not different in terms of p-values, but they also have to have specific confidence intervals, because we may remember that just because two things are not deemed statistically significant doesn't necessarily mean they're not different, because remember, it depends on the number of samples, right? The n value. So if you have really terrible, really low n values, really terrible variability, yeah, you're never going to include statistical differences, but that's what so the confidence interval will help you just really figure out how variable is that data, and it's associated with the power analysis, but we're not going to do a power analysis, right? Associated with the idea of it, what is the risk of falsely concluding that there's no difference, right? Falsely concluding that they're the same. We don't want to do that. Okay, so that's why we'll incorporate confidence intervals as well. So finally, if we have a statistically significant difference, does this mean the products are not interchangeable? Well, usually, say the vast majority of cases, yeah, they do have to be statistically not different. Okay, what I mean is if you the other way of looking at it is if you have p-values that show p-values under 0.05 usually, where there is a statistical difference. In most cases, they're going to be ruled not bioequivalent, definitely not bioequivalent, but then usually not therapeutically interchangeable. Okay, the FDA can make exceptions if the blood concentration is not that critical. For example, what if the Cmax is a lot more important than the AUC for like an antibiotic? Maybe the FDA can give a waiver. So these are not written in stone, but generally, right, you want to achieve no statistically significant differences for bioequivalence. Some of you, I imagine, are already thinking about this, so it's kind of I think it's an interesting concept. So it's convenient that like imagine you're making a new generic product. You say, well, I want to use a different dose than the reference product, and then you're like, well, I have an equation to allow that, so I could still show the FDA that I have bioequivalence even using a different dose. Is that always, is that always a valid approach? I guess is the question I have here. Right? It's convenient, but is it okay? So problems can actually arise when the AUC is not linearly related to dose. So imagine, right? So first of all, if this is true up top, that means you double the dose, you double the AUC, then that equation works perfectly. So that equation was designed assuming that there's a linear relationship with dose and area under the curve. So you double dose, you double the exposure to the drug. What if, as you get higher and higher and higher in the dose, what if the AUC rises more rapidly? So that's not linear. And why would that happen? What could happen, it's actually pretty rare, but it can happen in the case that what if, as you get higher and higher, some new mechanism is utilized, a transporter is upregulated, right? That increases absorption. And that's pretty rare. I would imagine much more commonly, as you get higher in the dose, you might start to saturate an absorption route if it's transport, like any drug that's ionic or charged. And this may, instead of going like this, it may actually go the other way, and you may saturate it, right? So either way, it doesn't matter. The point here is that if it is nonlinear, right, if it's curved on a linear plot, right, just a normal, even distribution on each axis, this, if this happens, we can't rely on that equation very well, right? However, if we know, like, if it's linear and up to here, well, maybe we can rely on it. But if the FDA knows, if they know that, well, once you go beyond this, that dose is no longer linearly related with the AUC, they may not allow that equation to be used. They may require you to actually change the dose, which would be the easiest thing to do, is to replicate the dose from the reference product data, rather than trying to fit an equation to this this curve. So again, if there's a summary there, a good rule, it would be to ensure that both formulations' doses are nowhere near the saturating absorption or elimination, actually, right? You really don't want to saturate any of those first order processes, okay? Because if a higher, and let's go back up a step, if a higher dose is saturating the elimination routes, the AUC will be exaggerated compared to the lower dose. And I forgot to mention that. I mentioned absorption being saturated, but what about elimination, right? Because the first thing I mentioned was absorption, but what about elimination, right? So we're talking about the whole AUC. Well, the whole AUC is determined by everything. It's determined by absorption, distribution, excretion, right? So any process there that is altered or saturated will have an impact on that curve, right? So again, the main rule of thumb is you cannot be saturating elimination, you cannot be saturating absorption, right? So you're going to stay within a range that we know is linearly related to AUC. The only way to know that is by testing it ahead of time to prove what dosage range is correlated in a nice, simple manner with AUC. So up until this point, I always mentioned area under the curve and Tmax and that kind of thing, and Cmax for measuring bioequivalence or bioavailability. Now, let me clarify, bioavailability is measured by the Cmax, AUC, etc. If we want to prove bioequivalence, saying two products are basically reaching the same effective level at the target tissue, basically it could be measured by other ways. Okay, so let me just clarify. If we look at the 4 bullet points down here, we can actually prove, like a drug company can prove bioequivalence usually by this method, plasma drug concentration, right? That's what I said, right, is the main one. So if the AUCs are the same between two products, or and the Cmax usually has to be both AUC and Cmax, usually that gives you bioequivalence, right? However, there are other cases where we can use other things. In vivo pharmacodynamic comparison. The onset of the pharmacodynamic effect could be lowering blood pressure, slowing down heart rate, So when does it start? What is the peak effect, right? And the total AUC of time versus biological response. You can actually do an AUC curve with just time and then the pharmacological or biological response, meaning change in blood pressure over time. Okay, so that is not direct. You're not really directly measuring bioavailability at that point. You're only measuring the pharmacodynamic response. Sometimes it is allowed. Generally, it's preferred to start with number one. Number one is more direct, right? But you're going to find out that some of these other things are acceptable in many cases. A clinical endpoint study, reduction in pain, etc. That might be the main. That might be the main measure we're using to show bioequivalence. Now imagine you might be able to imagine a product that, what if it doesn't get absorbed into the blood? What if it's treating a topical pain, right, skin or something like that? Well, it might be better to just look at reduction in pain rather than trying to measure the amount that got into the the dermis or something like that, or the epidermis, right? Rather than doing that, so we may have to go with a clinical endpoint rather than direct blood concentration. And then what about in vitro studies? Is it ever acceptable to just compare drug dissolution rate, outside of a human, right? Just in a beaker or one of those testing apparatuses that we talked about. Is that ever acceptable on its own? And the answer sometimes is yes, because if we know that the drugs are very well absorbed, if as long as they're dissolved, if we know they're very, very well absorbed, like 100%, and rapidly absorbed, they might actually allow that without doing the clinical trial for a generic product. So I'm going to break down the three alternatives to your standard bioavailability. These three alternatives that I mentioned, so pharmacodynamics, clinical endpoint studies, and in vitro studies, just on separate slides here with a little more detail. So in general, pharmacodynamic studies would make the most sense with locally acting drugs where you're not able to measure the drug concentration in the tissue, because you're not going to take biopsies usually from like a local tissue like the skin or something like that. So usually you measure the actual pharmacological effect rather than the drug concentration. I have to admit the first bullet here with reduced skin inflammation, that might be more of a clinical endpoint, but let me clarify that sometimes they actually look at skin color, reduced redness, which would be actually somewhat of a pharmacodynamic effect. And what I mean by that is what would be for most most cases the patients would be trying to achieve a lower pain in lower kind of swelling, but for the most part it's the pain that would be the clinical endpoint, right? Because that's what we're trying to avoid. That's our goal, is to reduce pain. Whereas the color of the skin, just reducing redness, might be more of just an indication that the drug has a pharmacological effect. That doesn't directly tell you, is the patient experiencing less pain, right? So it's a little very subtle difference, and this is not a good example, and I don't think I would ask anything like that on the test to tell me if it's pharmacodynamic or clinical. Bronchodilation from inhalers. Now, if you simply could measure the lung diameter or the bronchiolar diameter, that would be really pharmacodynamic, right? If you could do that with an imaging study, some type of imaging study live in a human, that would really be more pharmacodynamic. Whereas obviously clinical would be how many episodes of shortness of breath did the patient have, right? So that's the final endpoint. How are they feeling, right? Just like the things that matter, right? That matter to the patients. Are they breathing better or not? Whereas bronchodilation should make them breathe better, but it's not a direct measurement of that. So this is more of a pharmacodynamic measurement. Really clinical pharmacodynamics or pharmacology. Drugs that work in the GI tract might be better suited for pharmacodynamic instead of PK analysis because the blood level's not as relevant. So what I mean is, there are drugs you'll learn about that they're given orally, but they're targeting the GI tract. So measuring the blood concentration may not be relevant. And sometimes it's not very well correlated with the amount in the GI tract, believe it or not. So I'm trying to think of a good example. Drugs like Zetia, you might have heard of that blocks cholesterol absorption, ezetimibe or Zetia. That doesn't have very much blood absorption, but works in the small intestine. So you might, rather than measuring the drug in the blood, you might measure actual, you know, cholesterol absorption or cholesterol changes. So they might have to go with either pharmacodynamic or the clinical endpoint. Okay, so I'm trying to think of a better example. So if you had an antibiotic working in the GI tract only, which they do exist, oral vancomycin does not really get absorbed. You'd have to give it IV for a systemic infection. So if you were giving something like oral vancomycin to treat a GI infection, you might measure or culture the GI bacteria. Okay, so again, that's kind of a tough decision. Is that clinical or is that pharmacodynamic? So if there's a gray area, I don't plan on asking that, but basically what would be clinical there would be, is the patient having whatever their symptoms were, are they gone? Like, is there diarrhea or whatever they're getting from the infection? That's really more of a clinical endpoint. Okay, but again, there are gray areas there. So hopefully on this slide you'll see we have more real clinical outcomes. Okay, again, that's usually locally acting drugs because again, the systemic pharmacokinetics would not be that relevant. Okay, so why would we measure the blood concentration of a drug that's supposed to be treating a topical ailment, unless we were looking at toxicology, right? Which if we wanted to avoid systemic exposure. But our goal here, in bioequivalence studies, is to measure, is the drug reaching its intended target, right? So dandruff relief from shampoo, because how would you do a bioavailability study on a topical treatment of the scalp? How would you measure drug concentration on the scalp? Obviously whatever you put there is there, right? You put it there, so it's there. So it's not really relevant to measure the drug concentration in like scalp biopsies or something like that. So you would do the final endpoint, right? Is the dandruff gone, or is the patient having relief from dandruff, right? Dandruff is actually a microorganism, so that's what we'd be looking for. But not only, really primarily be looking for the patient to say, well, yeah, I feel better, I don't have any itching, I don't have any flaking. A duodenal ulcer, relief from an acid suppressive drug. Okay, so the patient would have less episodes, or hopefully no episodes, of gastric pain, right? That would be a good example of a clinical endpoint. Now you can go more in a gray area of pharmacodynamic by looking at the ulcer through an endoscopy, right? That wouldn't be as clinical, right, as the final endpoint. So again, that's the gray area. Acne relief from a topical cream, right? You pretty much will be just looking, is the patient experiencing less episodes of breakouts, less severe breakouts, right? That's a clinical endpoint. So please understand that, that again, usually pharmacodynamic and clinical endpoint studies, usually it's a local effect, right? Topical, we're not intending to get the drug in the blood intentionally, right? Finally, what about in vitro data? Like I said, if we have proof that the in vitro dissolution of drugs related to systemic absorption, we can model this accurately with an equation, we may be able to skip the in vivo studies. Remember I mentioned in a previous lecture in vitro in vivo correlation. So if we can prove that say for example, if 20% of the drug is dissolving, if I say that's always related to or associated with X% of drug being absorbed, and that's very consistent, right? A very consistent or predictable absorption based on drug dissolution, then we might be allowed to just go with drug dissolution rather than going into a human, right? So again, they might just study the dissolution profile of drug X or drug A versus drug B, and as long as they dissolve at the same rate, in the same extent of dissolution in the apparatus, maybe that's good enough sometimes, right? And they have to have really good permeability in the GI tract, and again, generally rapid absorption. So the biopharmaceutical classification system that we talked about would have to be really ideal there. So what are the actual rules for pharmacokinetic bioavailability studies? Okay, so there's some ground rules that are expected by the FDA. Okay, for again a clinical, this is not in vitro, this is a clinical pharmacokinetic study for bioequivalence. Patients should be randomized to product A or product B. It should be a crossover design, meaning patients who get product A will switch to product B, right? And usually there's a washout period where they're not getting the drug. So let me just give you an example. If they're maybe taking drug A for a few days or something like that, or even just a day, then they might give them, some time, maybe a few hours or a few days for that product to wash out and totally leave the system, and then switch to product B. And the reason why that's useful is because every single patient will have different physiology, so you can use, it's kind of like a pairwise comparison. So comparing one patient to themselves between these two different products will give you better statistics, right? It'll take out the whole physiology part of the equation, having variable physiology from patient to patient. So if they had poor absorption, if they had a bowel that was slower motility, well, that should hopefully affect both products equally, and that will be, then helping you in the statistics, reducing your variability. So you might be saying, well, for every patient, what was the difference between A and B? For each individual patient, what was the difference between A and B? And then you pull all that data together, the relative difference for each patient, and then what is the average relative difference, for example. They use both male and female participants, okay, because differences in physiology, for example. Testing under fasted and fed conditions, it's critical to understand if food will affect drug bioavailability. Usually they go with a high-fat meal in the fed conditions, to make sure that won't increase or decrease bioavailability. Two drugs, right? Two formulations, even if it's the same active ingredient, two formulations might have different bioavailability in the fed condition, and they might have the same bioavailability in the fasted condition, right? So it depends on the excipients and the way it's formulated. So you need to know both. Measuring the parent drug rather than the metabolites. So they don't usually use metabolites to measure bioavailability. Okay, they don't usually use the metabolites. So they're just measuring the drug that was administered. Okay, and obviously there are exceptions. If 100% of it gets metabolized through first pass metabolism, they may have to look at a metabolite in some cases. Cmax, Tmax, and AUC are always measured, right? If we're using our standard, standard bioequivalence, where we're looking at blood concentration rather than a local effect or topical effect. So these always have to be measured in those cases. Okay, so what is the maximum concentration? How long did it take? And then what is the total exposure, or AUC? So statistically speaking, when is bioequivalence achieved? So I alluded to statistics earlier. Now we're going to talk in more detail. And if this doesn't make sense immediately, I think the example on the next slide will help a lot. So the main requirements for bioequivalence are that, first of all, no statistically significant difference exists between the mean AUC and Cmax values between the two products. That means, both AUCs should have a p-value no less than 0.05. Okay, and usually they're going by, usually that's the alpha that they choose, saying that the p-value less than 0.05 means there is a difference. We don't want that difference, right? Same thing, if the Cmax values show a p-value less than 0.05, then that's a problem as well. That would immediately rule out bioequivalence. Remember, and this is going to require you to get a little bit, bring back your memory of statistics a little bit. And again, I took that a long time ago, and But this is one of the things that actually stood out, that you can not have significance in your p-value, meaning if your p-value could be higher than 0.05, right? But that doesn't guarantee you that there's no difference between these two groups. It might mean you have very high variability in your data, right? It might mean you just don't have enough patients, right, to give you statistical significance. So remember, what was the other thing in statistics that told us, that you have a strong enough study to be sure that there's no difference, or to be fairly confident that there's no difference between these patients? Again, the p-value doesn't tell you that. So you'd have to say, you'd use a power analysis, but in this case, what the FDA does is usually use, they always use confidence intervals, which is, it's related to the power analysis in a sense. So they always use confidence intervals. So just to remind you, a confidence interval on any value of data, so usually you're getting a mean value of something, right? You're plotting that, or putting it in a table. The confidence interval tells you basically that if you repeated that measurement multiple times, 90%, if you have a 90% confidence interval, then 90% of the time you would expect that number to fall within the range that you have there. Whatever range is given for the 90% confidence interval, 90% of the time the value you expect to fall within that range. Okay, so the way it works for bioequivalence is that first they look at AUC. If you divide the test product AUC over the reference product AUC, you want the 90% confidence interval, right, to be no higher than 1.2. That means the reference product AUC should not be any higher than 1.2 or 20% more than, I'm sorry, the test product should not be 20% more than the reference product. The test product should not be any lower than 20% or 0.2 under the reference product. That means you have to be close, basically close to the reference product in terms of your AUC and Cmax. But the confidence interval tells you how confident you could be in that data. So again, it's a little confusing probably at first, but I'll show you an example. But this means the 90% confidence interval for the test product pharmacokinetic values is never exceeding 20% more or less than the reference product. Okay, and this is what it looks like. So on this slide, this is hopefully going to be a little bit better to make sense out of it. So imagine we have a test drug and a reference drug given at the same dose. The AUC is just, we don't need units right now, but at 2708, 2718, the Cmax is 345, 357. The ratio of the test to reference, this is just dividing, right? You're just dividing right now. So test divided by reference, 99.6. Test divided by reference, 96.6. What I did was converted this fraction into percent by just multiplying by 100. And in this data here, this confidence interval data, I'm not giving that. I mean, I'm not asking you to calculate that, and you couldn't calculate that from this, right? You'd have to at least get the number of patients and the standard deviation. So please don't try to figure out how I came up with this. I just made it up. Okay, so if it's, for example, 95.4 to 103, right? So our answer was, our actual ratio is 99.6. We're saying that we're basically 90% confident, or that 90% of the time, if we repeated this experiment or these measures, 90% confident that we would get between 95.4 and 103 for the ratio. Right, and that is actually acceptable because it's above 80 or 0.8, right, and it's below 120, right? Above 80, below 120, that's good. Cmax, 89.5 to 112. We're above 80 on the lower. There's a lower end of the range. We're above 80. The higher end of the range, we're below 120. That's good as well. So the confidence intervals are good. It tells us there's bioequivalence. Always look at the p-values. Those have to be higher than 0.05, and they are in this case. So that means there's no statistically significant difference here. The confidence intervals are in the appropriate range. Okay, so bioequivalence has been achieved, and that's all it is. So the wording on the previous slide makes it more complicated. When you actually look at what's done, it's not that bad. Okay, so it means there is some leeway, right? There's a little bit of leeway in right, that a drug doesn't have to be superimposed perfectly on top of the reference product, right? It can have some variability, but it's got to be somewhat similar in not only the rate of absorption, right? The rate of absorption meaning related to C max, but also the total absorption, which is related to the AUC. Both those have to be pretty similar. So hopefully that makes a lot more sense now. So if you like statistics, you might not need to hear this over and over again. I apologize. If statistics kind of wasn't your thing, and I understand that, it's a lot of people don't really enjoy statistics, and that's understandable, like I said. But I wanted to mention this one more time because I think it's really important to get this. Why do we need both the p-value and the confidence interval? Right? Again, failure to find a significant difference, maybe because of high variability in the samples or low sample number. Okay? You should never, and honestly, I see scientists do this constantly, right? When I go to conferences, when I talk in meetings about science, there are so many scientists that have so much confidence because in their data that there's no difference because their p-value is higher than 0.05. That just tells you, right, that you can't be confident that there is a difference, right? It doesn't guarantee you that there's no difference, and that means that right, you could easily achieve no statistical difference if you just have super high variability, right, in your data. Right, so if you have really high variability, you'll never get statistical significance. Does that mean though, if you had a lot more samples, if you had like 1,000 patients, does that mean that you wouldn't find a difference? No, it does not mean that, and that's why you do a confidence interval analysis. The confidence interval tells you, can you be confident that there is no difference? Right? Can we be sure that there's no difference? And the answer is never absolutely, right? Because it's a 90% confidence interval. It's not a 100% confidence interval, right? Because really, you would never achieve, right, the 100% confidence intervals. How could I put it? We have to have a judgment call, right? If we were trying to get 100% confidence intervals to overlap perfectly and not show a difference, that would probably never happen because you would need probably the whole population on Earth to get good enough data. So the point is we have to make a compromise and say, well, what is good enough to say, well, these probably aren't that different, right? So even though it's all numbers, someone made a judgment call that 90% was good enough. Someone made a judgment call that 0.05 was good enough for the p-value, and that was statisticians many, many years ago, decades ago, right? Again, there's in any objective value, there was some kind of subjective point at which we say, well, that's good enough, right? That's a judgment call. So for example, if two plasma curves were on top of each other and bioavailability looked identical, but the standard deviation were very high, we might not be able to conclude that they're bioavailable, right? Because we don't have enough samples. So I have that coming up here. So imagine in both examples here, the mean plasma concentration is nearly the same at each point, right, in both examples. Of course, the p-values would be higher than 0.05. What it means, there's no statistical difference when we look at the p-values, right? Even with this data or this data, right, you're not going to get a difference, okay? But our confidence that there is no difference is higher for the data on the bottom. The error bars are much tighter, the standard deviations are much, right, smaller. So even without knowing a lot about statistics, but you know what standard deviation is at least, right? Looking at this, so well, I'm already more confident that these numbers are similar to each other in reality, right? If we've repeated the measurement more and more and more, I'm confident that these would stay about the same place here. This one I'm not confident that if we repeated these measurements that they would stay right on top of each other. So that standard deviation is directly related to the confidence intervals. So the confidence intervals here might be much broader, and here they might be much narrower, right? So keep that in mind, and that's why p-value is not enough on its own, right? Just because it's over 0.05, that doesn't mean enough on its own. This slide is just again to remind you what is the purpose of each of these measurements. Of course, Cmax for peak drug exposure. Tmax is directly related to the rate of absorption, and AUC is the total drug exposure. We would love for a new formulation to be bioequivalent and match all these, right? The bioequivalence will be shown for all these different measurements. Sometimes the FDA, though, will allow one of these to be substantially different if it is not expected to be clinically meaningful, right? So even though I said that we have to show that there's no difference between all these, right? Really, I said Cmax and AUC. Again, nothing here is written in stone. If you know for a fact that the peak drug concentration is not that important, for example, a drug that's not toxic at all, or very low toxicity, and its effectiveness doesn't depend on the peak, then maybe we don't care about that, right? Maybe we don't care about Cmax, something like that. You never know, right? So it all depends on the drug that we're looking at. On the other hand, if the drug has a very narrow therapeutic window, in almost definitely, in all cases, we're going to say all these are probably really important, right? If a drug is very, toxic, we definitely want to know how high does it get, what is the total exposure, and then how long does that take. That's all going to be important then. So just keep that in mind. The FDA has the ultimate say in a lot of these things. So we're in the home stretch here, and I just wanted to talk about the different levels of equivalence and the different terminology here. So, the FDA has come up with a lot of different terms, and they've been out there for definitely decades, at least. So these terms are really just telling you how similar, in a sense, are the two different products if you're comparing them. So this would be beneficial, for example, a pharmacist or a physician or anyone that's having a say in the choice in drug therapy to say, are these products therapeutically equivalent? Are they just pharmaceutically similar but they haven't been deemed therapeutically equivalent, et cetera, et cetera? I'm going to break these down into what each one means. And there may be cases where, for example, what if a physician wanted to know, well, okay, so drug A and drug B are not therapeutically equivalent, but they're, for the most part, otherwise they're similar. So maybe I can, as a physician, right, you're allowed to make that decision. The FDA doesn't tell you as a physician, or again, if you have prescribing rights in a state as a pharmacist, right, if you're saying, well, I'm going to use another product, even if it's not therapeutically equivalent. Well, if if it's at the level of the prescription, and I don't mean, if you're working, in community pharmacy and the prescription's already been written, you're not going to be able to change that. But I'm saying that, when it's first written, I'm going to use drug A versus drug B, and they're not therapeutically equivalent. It might be helpful to know that at least they're pharmaceutically equivalent, and you may need to change the dose, but there's some level of similarity between those two products. The FDA, if you go to the website, you'll see they're not intending to tell a prescriber that they cannot use one product versus another product. The purpose is at the level of the pharmacist, to say, well, what is allowed to be substituted without consulting the prescriber, right? Which ones are allowed? So let's break these down though before I talk any more about it and show you what I'm talking about here. First is pharmaceutical equivalence. Okay, pharmaceutical equivalents are drug products in identical dosage forms and routes of administration with identical amounts of active ingredient in the same form, same salt. Okay, so that means they're both, for example, it could be both tablets intended for oral use, the exact same amount of active ingredient in the exact same salt. That means metoprolol tartrate in both of them. Solid, tablet, same route. So far they look the same. They must match each other in terms of disintegration and dissolution. Content uniformity, standards of quality, everything is the same except they can have slightly different, well, really any different shape, but it still has to be, again, in this example, it would have to be the same consistency or tablet, same type of solid. You could vary the shape, the scoring, so the line for breaking it, the packaging, and the excipients can vary. But guess what? Bioequivalence is not part of this. So if I say two drugs are pharmaceutical equivalents, it means everything is pretty much the same except we have not studied the absorption, right? So, if you're a prescriber, right, the only thing you don't know is, if you give equal doses of each drug, will the patient experience the same blood concentration? But you can probably have some confidence that, right, they'll be fairly similar. But again, that hasn't been studied. So just understand that bioequivalence is not part of pharmaceutical equivalence. Okay, so you're not studying the actual absorption. Pharmaceutical alternatives. This is a looser one, meaning the products are less similar. So the drug products that contain identical active ingredient, or could even be its precursor. So for example, something that is a prodrug. So product A might be the active ingredient, the active compound. Product B might be a prodrug that's converted to that, right? But they're not necessarily in the same amount or dosage form or salt or ester. That means they're alternatives because they're not the same, but you think they'll probably produce a similar effect in the patient. Example, tetracycline hydrochloride versus tetracycline phosphate, right? Of course, they're both the same antibiotic molecule, but they're a different salt form. So they might be absorbed differently, they might have different stability, right? They will never be pharmaceutical equivalents. As soon as you add a different salt, they are never pharmaceutical equivalents, right? They could easily be pharmaceutical alternatives, and these ones definitely would be because it's the same molecule, again, in a different form. Extended release versus immediate release, they're never equivalents, but they are alternatives, right? Because they're not going to produce the same absorption, right? But keep in mind the pharmaceutical equivalents and pharmaceutical alternatives, they're not really studying absorption at all, right, in these cases. The main thing they're focusing on is what is the active ingredient, is it the exact same salt form or is it a different salt form, and that kind of thing. Therapeutic equivalence, now this is more stringent, meaning the products are much more similar to each other. So pharmaceutical equivalence for which bioequivalence has been demonstrated and can be expected to have the same clinical effect and safety profile. For this definition, therapeutic equivalence must contain the exact same ingredient, and I don't have it here, but of course it would be the same salt form. Okay. So example, morphine and codeine cannot be therapeutic equivalents, even at equally effective dosages, right? Because they are totally different active ingredients, even though codeine is metabolized to morphine, again, because they're different active ingredients. Therapeutic equivalents have to, this is the critical point here, have to be bioequivalent. This is the only type of equivalence that requires bioequivalence, right? So, and let's think about this. So that's the ultimate level of evidence, right? They're absorbed the same rate and extent. That's the bioequivalence. But then you back up a step and say they also have to be the same salt form, right? Same active ingredient, but the same salt form. So basically, it's everything has to be, as far as we could tell, the same, right? Same dosage form, same active ingredient, same, like I said, same salt form, same absorption profile, Cmax, Tmax, AUC. So therapeutic equivalents meet the highest level of stringency with respect to, can you substitute? So of course, if a pharmacist is substituting a generic product for a brand name product, they generally, they would have to have therapeutic equivalence, right? In almost all cases, right? Unless there was an emergency in the government announced that temporarily we could suspend this. But for the most part, they have to be therapeutic equivalents to be substitutable. So a little while back in this lecture, I mentioned the Orange Book. We're going to come back to that briefly now, and then we'll end the lecture. The Orange Book, remember, basically lists reference and generic drug products and tells you, are they equivalent or not, basically. So again, now that you understand bioequivalence, we'll be able to look at the way drugs are listed in the Orange Book to determine whether they are allowed to be substituted. Just remember, the FDA can give special waivers. Even if drugs are not perfectly bioequivalent, they may sometimes still be considered therapeutically equivalent, again, and then that would mean able to be substituted legally for the reference or brand name drug. Okay, so the good thing is we have now it's online database. Of course, it used to be just a book decades ago, but an online database that tells us without any kind of mystery, right? Can we substitute it or not? Right? There shouldn't be much of a judgment call. It's just yes or no, right? Is the FDA going to allow substitution of a generic for a brand name, or or one generic for another one, basically? So this table is an example of what you would see in the Orange Book. First of all, I want you to be again, first and foremost, familiar with the major codes. So what this means is that if a drug has this code A, right, and this, you'd be assuming right now we're looking at a generic product. Usually the reference product is the original brand name product. A tells you, if it's next to that generic, A tells you that is equivalent. It's equivalent to the reference product. What these mean, again, they should all be A as a starting point. That's the main code. But then A what? So A means there's neither actual nor potential bioequivalence problems. Oral solutions. Okay, so that in some cases it may mean that they didn't actually study the bioavailability, because it's clear that the oral solution should have the same absorption. But the main thing you need to know is that the A means they're equivalent. That means substitutable, right? So these are drug products that are therapeutically equivalent. So you should be able to quickly look at this if there's a new generic product and say, well, is that able to be substituted for the brand name? And say, well, it has an A, so the answer is yes immediately. But what do these other things mean? So AB means actual or potential bioequivalence problems have been resolved through testing. That means not only are you saying that there shouldn't be a problem, you're saying we've proven that there is no problem through a clinical test. Whereas the first one means they probably didn't even do the bioequivalence study because they don't expect any problem whatsoever because it's an oral solution. So AB means it's actually been tested and there's no problem. AN, and now we're getting down to different formulations. AN means products for aerosolization or nebulization. The way I always remember it is the N. A nebulizer is for inhalational use, right? It's I don't know if you've seen those yet, but they're those kind of larger machines, and they've probably gotten smaller over the years, that they produce a mist that you breathe in. So I always connect that N with that N for the nebulization there. So you'll hopefully always remember that. So AN. AO, injectable oil solutions. So always look at the O and connect that to the oil, or the O and the oil. AP, injectable aqueous solutions. So I look at this as, and it's a little possibly confusing to memorize all these this way, but P, parenteral, but it's parenteral and aqueous. Okay, and so there's no P in that to help you memorize that. There's no P in that description, but I think of it as the opposite of, right, the AP is your opposite of the oil because it's aqueous, AP is your standard injection, right? Your standard parenteral dosage form, which is most of them are not oil-based or oleaginous. Most of them are aqueous. So AP, just standard parenteral. AT, topical products, right? So it all makes sense. For the test, I want you to know what all the A codes mean, okay? Please know what all the A codes mean. And then, again, what the overall A means is just, yes, you can substitute them. Overall, you need to know that B means, for the first letter, B means you could not substitute them, okay? And there's a lot of reasons, and actually I don't want to ask you about that. I want to ask you what the A codes mean, especially the first and second letter. First and second letter. What do those each mean individually? And then just know that B means, well, I guess I just made it up now, but you can say bad because they're not equivalent, right? They're not therapeutically equivalent. So, right, you say it's a lower grade, or you could think that, or you could think just B for bad, but they're not equivalent. So, what do they mean? You don't have to memorize these, but further investigation required. Extended release products are usually not going to be your bioequivalent. We might have obvious documented problems with bioequivalence. Delayed release oral formulations. Okay, so that says delayed, I'm sorry. But delayed is different from extended. Delayed means the onset of absorption is actually later as well. Again, BN is nebulized. BP is, I know it's possibly confusing compared to the AP, but BP means potential bioequivalence problems. BR means rectal administration forms. BS, drug standard deficiencies. Maybe the purity is not appropriate in the generic product. Again, maybe it's an impurity, or the manufacturing method might be inadequate in terms of the quality controls. And then BT is topicals. So again, memorize for the testing purposes. Memorize all the A codes. Okay, and then the basic A and B. What do they mean for the first letter? And that's it. This concludes the lecture on bioavailability and bioequivalence. I really strongly encourage you to go back to the learning objectives and see if you could answer those now without looking at the notes. And if not, please go through those again. Okay, and again, hopefully you found this interesting and useful. I know it's directly related to some of the law material that you'll have later on. So keep in mind this is going to be really important for your future. And again, thank you for listening.